There are so few aneuploidies of the autosomal chromosomes that are known, because almost all of them are fatal in embryonic stages. We have many named aneuploidies of only a fragment of an autosomal chromosome. For example, cry of cat syndrome, infants with cry of cat syndrome, which has been a named disorder for over 100 years, originally described by a French doctor. Individuals who, this is chromosome number 5 here, individuals who have this aneuploidy are only missing a piece of one of their chromosome number 5s. Why would this one spot here be a common breakage point? Well, it turns out it's a crossover hotspot for during crossing over during meiosis. So it's a common place for a piece of chromosome to get lost. This is also not survivable past infancy. Babies that are born with cry of cat syndrome generally survive days or weeks. It's severe. So even though we don't, monosomies of the large chromosomes here, the autosomal chromosomes, are not known because they're not survivable, we do have named aneuploidies of just pieces of them. Nondisjunction of the sex chromosomes is a completely different story. So we'll talk about both X and the Y aneuploidies. So if nondisjunction occurs with the X chromosome, we can end up with female embryos that have extra X chromosomes, or only one. If there is an egg that is missing an X chromosome, we can end up with the 0 there. That means no, or I think that's a letter O. That means no chromosome is there. So they just have an X, one X. If it's a male embryo, we can end up with males that have extra X chromosomes. If there's no X chromosome at all, that's not viable. So having only a Y chromosome and no X is incompatible with life. There's just too many genes on the X chromosome. So surprisingly though, trisomy of X is very hard to detect because these individuals are almost completely phenotypically normal. Turner syndrome is the most severe of these three. And it's even not that severe. Klinefelter syndrome also almost undetectable. Most males who have Klinefelter syndrome don't know until they're an adult that they have it. So triple X females are completely healthy. The only thing that's different about them is they tend to be a little bit taller than you would expect based on their family, because there happens to be a height gene on the X chromosome, one of the genes that contributes to height. And so triple X females have an extra dose of that height gene, and so they tend to be a little bit taller. But other than that, no significant problems. Klinefelter syndrome, that's males who have 2 X chromosomes and a Y. Now, because they have a Y, they have the SRY gene. That's going to turn on male development in the embryo. And as well, Klinefelter syndrome, males are almost completely normal. They tend to be infertile, and that's generally how they find out that they're Klinefelters, because the 2 X chromosomes act to disrupt the development of internal organs. Monosomy X is the most severe of these aneuploidies of the sex chromosomes. If you have only 1 X chromosome, that's Turner syndrome. They are sterile. You generally, it can go undetected until school age. They're going to be developmentally delayed, shorter in stature, again, and that's because there's a height gene on the X chromosome, so they only have 1 dose of that. This is very mild. Turner syndrome is, in general, milder than Down syndrome even is. So aneuploidies of the sex chromosomes tend to be much less severe to being completely insignificant, like the triple X females, which are even fertile. It doesn't even affect fertility. Here's a little video of, oh, I got to go back to my regular pointer here. to start this video. So here's a video of nondisjunction during mitosis, but what you'll be able to see is clearly a little chromosome pair that is off track and ends up not being attached to the spindle. The sister chromatids are still going to separate, but they both end up in one of the daughter cells. So one cell is going to end up with an extra chromosome, and the other daughter cell is going to be missing a chromosome. So prophase, prophase, late prophase, or prometaphase. You can see that one little chromosome is not getting in there. It still separates. Let's watch that one more time because it's short. Okay, we're in prophase, prometaphase. Chromosomes are working their way to the metaphase plane. Separation, one little chromosome gets lost. So nondisjunction does occur in mitosis, especially in cancer cells. Cancer cells often have extra chromosomes and missing chromosomes. Nondisjunction of the Y chromosome. So this was discovered many, many decades ago, that some males have extra Y chromosomes. And although people have looked very, very hard for something negative to associate with extra Y chromosomes, in fact, they are phenotypically normal, other than, again, being a little bit taller than would be expected based on their siblings. So if you want to find XXX females or males with extra Y chromosomes, scientists look where you find lots of super tall people, like the women's Olympic volleyball team or the NBA for men. So people tried really hard. I don't know why people tried so hard for the Y chromosome to find something negative associated with extra Y chromosomes. They sure didn't do that for the extra X's for females. People looked at IQ, they looked at criminal behavior. There was a lot of research into that, and in fact, it all turned into a big nothing. In fact, there's no greater association of anything really with the Y chromosome other than height. Men with extra Y chromosomes don't have more testosterone or anything like that. And it's fairly common. About 1% of males and individuals with extra Ys, in fact, are fertile, so there's no way they would even find this out under any normal circumstances. So why is aneuploidy of the sex chromosomes not really consequential at all, except for Turner syndrome, really? And the reason is because of something called dosage compensation. So for all of the autosomal chromosomes, if you have an extra one, that means that you have 50% more of everything that the genes on that chromosome makes. So that's what causes the abnormalities that we see in Down syndrome. So individuals with Down syndrome, all the genes that are on chromosome 21, they have 50% more of a higher dose of everything that those genes make. If you have a monosomy, so if somebody's missing one of the autosomal chromosomes, they're only going to have half of whatever those genes make on that particular chromosome. And that's what causes the lethality for most of the autosomal aneuploidies and the problems that we see in the ones that are survivable. So the X chromosome is different, though, because in mammals, there already has to be a way to deal with the different dose of X chromosome, because we have some individuals that have 2 X chromosomes and other individuals that have 1 X chromosome, and the X chromosome has a lot of genes on it. So our bodies already have a way to deal with that, and that's called X inactivation. So in cells that have 2 or more X chromosomes, all the X chromosomes are inactivated except for 1. And that inactivated chromosome is in fact visible, and it's called a bar body. And we can see, where's my pointer? That bar body is visible in the nucleus of cells. So here's the nucleus of a female cell, a mammalian cell, probably it's a human cell, and we can see the bar body there in the nucleus. And in fact, we can see this inactivation in cats. Cats happen to have a fur color gene on their X chromosomes. And so female cats that have one X chromosome has an orange coat color allele, and the other X chromosome has a black coat color allele or a brown coat color allele. They're going to be a mixture of cells that have inactivated one or the other X chromosome. So if you know anything about cats, you probably know that calico and tortoiseshell cats are female. And that's because what we're seeing in their coat color is X inactivation. So where their fur is orange, that's where the black containing X chromosome has been inactivated, and where they're black, that's where the orange containing X chromosome has been inactivated. So theoretically, if we ever found a calico cat that was male, they would be a Klinefelter syndrome cat. And in fact, such a cat has been found in California a few years ago at an animal shelter. There was a calico cat that was a male, and luckily somebody working at the shelter realized what this meant. And a scientist at a university bought the cat and adopted it as and in fact did the DNA test and showed that in fact it is Klinefelter's male with 2 X chromosomes. So tortoiseshell, this is tortoiseshell cat. So tortoiseshell and calico are the same, except for calicos have a white patch. They have some white on their face and their chest and their belly. That's a different gene somewhere else. Tortoiseshells are still the mixture of black and orange or brown and orange. They just don't have the white. So 2 X chromosomes, one with an allele for black and one with an allele for orange. And there's a patchwork of inactivation. And now this inactivation occurs early in embryonic development. So that's why we get these irregular sized patches. Now, human females have the same patch mosaic work of inactivation. We just can't see it because there aren't any skin color genes on the X chromosome. If we could see it, those patches would be about the size of a quarter. Dosage compensation. So here we have so why isn't it, if we have this dosage compensation, if one of the X's is inactivated, why aren't Klinefelter's males completely normal? As I said, they're almost completely normal, and most males who have Klinefelter syndrome don't find out until they're adults, and they find out that they're infertile, and that's generally how they find out that they have Klinefelters. And there's 2 reasons. One is that there is a piece of the X that's not inactivated. There's a piece of the X and the Y chromosome that behave like a little pair of homologous chromosomes. It's called the pseudo-autosomal region. That part of the X is not inactivated, and crossing over actually occurs there. They pair up during meiosis. And also, as has been discovered, X inactivation isn't 100%. So even though we can see this smashed up little X chromosome here in the bar body, it's not 100% complete, and there are genes on the X chromosome that are not inactivated in either copy. And so that is what makes the difference. And some of these genes are involved with embryonic development of the uterus and fallopian tubes. So these don't get inactivated. And so that's why Klinefelter's males are not completely normal. So aneuploidy of the X still has some effects, although it's undetectably mild in females who have extra X chromosomes. So here's some that have been found. So here's a normal male. We don't see any bar body. And here a fluorescent tag has been used to find the bar bodies. So these are just the nuclei. So in a normal female with 2 X chromosomes, we see 1 bar body because all the X's are going to be inactivated except 1. So if there's only 2, 1 bar body. Triple X female, 2 bar bodies. 4 X's, 3 bar bodies. 5 X's, 4 bar bodies are going to be found. And Turner syndrome Female, no bar bodies because she only has a single X and no Y. We talk about some genes being linked. So if genes are linked, that means that we're not talking about different alleles of one gene, we're talking about different genes. Every chromosome has hundreds to thousands of genes, and those genes are linked because they're physically attached to each other on the same piece of DNA, so that generation after generation, those alleles that are on the same piece of DNA are going to tend to be inherited together. We know now that those linked genes can be separated by crossing over during meiosis, but that was not initially apparent to early geneticists. So Morgan started figuring out what was happening with linked genes when they first started identifying fly mutants. They identified linkage groups, and 4 linkage groups were identified in flies, and that's because that's how many unique chromosomes there are in flies, is 4 unique chromosomes. Their N number is 4. And for example, so here is 2 of Morgan's mutants that he found. One mutant, and again, like we saw before, the name of the gene has something to do with the first mutant of that gene that was discovered. So we have a pigment gene that's called black body, and the B that symbolizes this gene is because that's the first mutant of this gene that was discovered, and then the B plus indicates the normal gray body allele, which is dominant. And then we have VG is for vestigial wings. So again, the name of the gene is related to the first mutant that was identified in VG+. That's the normal size fly wings. So vestigial wings means these flies can't fly if they have vestigial wings. Their wings are too small. And these are both recessive traits in fruit flies. So when we cross our normal wild type fly with one that is homozygous for both of these recessive traits, think of Mendel's P experiments, except we're using 2 traits at once, which is a dihybrid cross. So now we have our F1 generation here, this female who's going to be heterozygous for both the black body and the vestigial wings alleles. And then we're going to do a test cross with another recessive individual. And what this is going to do is this is going to allow us, because this recessive individual has nothing but recessive alleles, it's going to allow us to see where the dominant alleles go in the next generation. So, so here we have possible offspring here. So all of the eggs from this female are possible eggs here at the top. So we can have eggs that have the 2 dominant alleles, that have the 2 recessive alleles, and that have 1 dominant and 1 recessive. And then the sperm are all going to be the same. They're all going to have the black body and vestigial wings recessive alleles. So if these genes are not linked, if they are not on the same chromosome, then we would expect these traits to sort independently, because remember, the chromosomes sort independently during meiosis. So if they're on different chromosomes, that means that all of these are equally likely, because all of these gametes are equally likely. If, however, they are on the same chromosome, then they are linked. And that means the only way that we're going to get these recombinant gametes is if crossing over separates them. So the number of these that we get is going to be important for determining linkage. So if they're on different chromosomes, we would expect a 1 to 1 to 1 to 1 ratio of offspring here. If, however, they're on the same chromosome and there's no crossing over, then we would expect to only get the parental types, only gray body and normal wings and black body vestigial wings if they're on the same chromosome. However, that's not what Morgan and his students found. In fact, you get a smaller number of these recombinant flies. And what that says is that even though these are on the same chromosome and they stay together most of the time, sometimes they don't. And this is decidedly non-Mendelian, these kind of results. So Mendel picked traits that look like this when he crossed them. Mendel's 7 traits that he looked at in peas. When he did dihybrid crosses, he got all these kind of numbers, which is because almost all of his traits, all of his 7 traits except 2, were on different chromosomes. 2 of them happened to be on the same chromosome, just really, really far apart. So Morgan and one of his students who later worked on it after Morgan did, realized what this meant, that linkage groups, first of all, linkage groups were an indication that genes are on the chromosomes, because remember when Morgan started doing his experiments, they did not know for sure that genes are on the chromosomes. And it also indicated something else, that not only were these genes on the same chromosomes, but that somehow these chromosomes were unstable and that there was recombination occurring with a small percentage. And some genes, there was more recombination, some genes less. And this became really important for mapping the chromosomes of Drosophila and other species. So here's the same exact thing, but with 2 of Mendel's traits that he used. So we have the dominant traits, yellow and round, and the recessive traits, green and wrinkled. So if we do the same thing, we have a hybrid parent that has 1 dominant yellow and 1 recessive green, 1 dominant round, 1 recessive wrinkled, and we cross it with a homozygous recessive parent, which would be the wrinkled green. This is the same kind of experiment, but with plants. So if these 2 genes are on different chromosomes, if they're not linked, which Mendel found they were not linked, then we would expect all of these offspring to be equally likely. If, however, they are linked, then we should get fewer of these recombinants that have 1 dominant and 1 recessive. And that's because originally, going back to the parent that we used to make this hybrid, the dominant alleles were on the same chromosome and the recessive alleles were on the same chromosome. So the only way that we can get these recombinant offspring is if something happened to those chromosomes and the strands were broken and recombined. And so Morgan actually proposed, and this was before it was known the mechanism of inheritance was in fact DNA. This was before they knew how DNA replication occurred or any of that stuff. Morgan, with these linkage groups, and the fact that linkage was incomplete, that somehow these chromosomes were unstable and you get some recombinant phenotypes, he proposed that the physical connection between the genes was breaking and crossing over somehow must be occurring. And people thought this was nuts, which, you know, of course, ideas like this, often people think that. But it, you know, Morgan turned out to be correct. And here's a nice transmission electron microscope image where you could clearly see the 4 chromatids and in fact the crossing over that is occurring in 1, 2, 3, 4 different spots here on this chromosome. So back to the flies. So here we have our gray body normal wings and our double recessive black body vestigial wings. So this fly is heterozygous, so the light pink chromosome, that's representing the one that has the recessive alleles on it, and the red one is the one with the 2 dominant alleles on it. So when meiosis occurs, the only way we're going to get these recombinant individuals is if these chromosomes do not stay intact. And in fact, we know that crossing over occurs, and in flies it's random where the crossing over occurs. And so occasionally, not every time, but occasionally during meiosis, crossing over is going to separate these alleles and create a new chromatid that now has one recessive and one dominant allele. And that's where we get these recombinant individuals. So when these combine with the recessive allele containing sperm, we end up with parental type, and recombinant. And these, however, are going to be less frequent. So we're going to get mostly parental types and a smaller percentage of the recombinants. And they realized that they could use that. Let me go back for a second. They realized that they could use these percentages to map the chromosomes, because the further away these genes are from each other, the more likely crossing over is going to separate them. So the higher percentage of recombinants that you get, the further away the genes are from each other. So this crossing over vastly increases the amount of variation that you get in the offspring of sexually reproducing species. So we already talked about some of the things that increase variation, like new mutations. New mutations are the raw material for this variation, and sexual reproduction itself increases variation by creating millions of possible gametes every generation and tens of millions of possible zygotes. And this gives natural selection more to work with. And there must be a strong evolutionary advantage to this, because it has been preserved in almost all eukaryotes. It's the exception eukaryotes that do not have sexual reproduction and don't produce this huge variety of offspring. So one of Morgan's students, Alfred Sturtevant, he was the first one to really map a chromosome, and he used this crossing over percentage in order to do that. So the further apart two genes are, the more likely that crossing over will occur. And this worked because in flies, where crossing over occurs is pretty much random. That would not have worked if he had used mice, for example, because in mammals, crossing over mostly occurs at crossover hotspots. It tends to occur in the same spots over and over and over again. And it would not have worked as well to map the chromosomes that way. And now that the Drosophila genome has been sequenced, we can see that Sturtevant was correct, at least in the order of the genes. Some of the distances, approximate physical distances, were off a bit, but the order of the genes on the chromosomes he was able to determine correctly using this method, which was tedious and took years and years, and the discovery of all these mutants, and now we can sequence a genome in a few days. So other than we already talked about some of the non-Mendelian traits like polygenic traits and ones that have an environmental factor as part of the phenotype. But there are some other exceptions to this normal type of Mendelian genetics. And one is genes that are not in the nucleus, which for us is mitochondria. For plants, that would be mitochondria and chloroplasts. And also some that are genes inside the chromosome, silencing of genes through tagging of those genes that is not erased every generation. So the organelle genes, we mean mitochondria and plastids, especially chloroplasts. So if you remember way back earlier in the semester, we talked about the origin of eukaryotes and the fact that mitochondria and the plastids, like chloroplasts, were once free-living bacteria, and they have bacterial ribosomes, they have bacteria-like chromosomes, although their chromosomes are very small. So human mitochondrial genome has less than 20 genes, whereas a typical free-living bacteria, has 2,000 to 3,000 genes. So they have a very small genome, but nevertheless, they have DNA. And the inheritance of this DNA and the genes that are associated with them is maternal, almost 100%, exclusively through the maternal line. And there are genetic defects and genetic disorders that are associated with the mitochondria. And because the mitochondria are involved with aerobic respiration, you can imagine that these diseases are primarily about energy consumption, and they especially affect the nervous tissue because our brain and our nervous tissue cannot ferment. So they have to use oxidative respiration for their energy. They have to use their mitochondria. So mitochondrial defects are going to be more severe in nervous tissue and the brain. So this inheritance, though, is going to be a little bit different than other types of traits because 100% of the mitochondria that an individual inherits are from their mother, and it doesn't recombine. It's exactly the same. So we can trace back mitochondrial DNA for dozens of generations, and the only changes in the mitochondrial DNA will be new mutations. There's no recombination or adding or subtracting or anything like you do with sexual reproduction in the nuclear genome. So that means that if a female has one of these mitochondrial disorders, she has a 100% chance of passing it on to her offspring. So all of her children will inherit it. If a male has one of these mitochondrial disorders, 0 of the children will inherit it. So different kind of inheritance pattern than we've seen before. And because of that, a solution to this has started to be used, has been legalized in the UK, and there's actually been at least one baby born in the UK using this method. It's been now approved in the US, but I have not heard if any babies have been born using this method. So the question is, okay, we have this woman here who has one of these mitochondrial disorders, and they can get worse in the next generation. So she might be okay, but her children might be even worse than she is, sicker. And 100% of her children are going to inherit this mitochondrial disorder. If the husband has it, it doesn't matter because he's not going to pass it on. So, but it's only the mitochondria that are the problem. The nucleus is okay. So what they're doing now is you need a third person to donate healthy eggs. And we take the donor's eggs and remove the egg nucleus and then replace that nucleus with the nucleus from a newly fertilized oocyte from the couple. And that way you get the healthy DNA or in the mitochondria of the donor and the nucleus from the zygote that these two made by in vitro fertilization. And it works pretty well. The biggest problem is when you pull this nucleus out. If you remember back from our cell anatomy section back in the beginning of the semester, around the nucleus is this spider web of microtubules and intermediate filaments, and when you pull that nucleus out, you drag with it all of these fibers and other things, including mitochondria. So when you put this nucleus into the other egg, you do get some mitochondria from this mother who had the mitochondrial disorder. So in offspring that result here, you get about 4% of the mitochondria are going to be the unhealthy mitochondria from the biological mother. So, and it's a question was, is that going to be harmful? Is it going to be harmful to have 4% of the mitochondria from a different individual? And we do know that when you have mixed types of mitochondria in one cell, that alone can cause problems. So they're going to be watching any babies born from this very carefully to see if they are healthy or if they show mitochondrial issues because of the fact that even though most of their mitochondria are healthy, they have 4% of the defective mitochondria, and they also have a mixture of 2 different kinds of mitochondria in one cell, which we already know that can be bad as well. So this is, well, the first baby that was born with this method was in 2018. So hopefully we will have news at some point about how these babies are doing that are being born with this method. So when they say 3 parents, that's a bit of a stretch. So the only DNA from the 3rd parent is the mitochondrial DNA. Another type of genetics is epigenetics, and this is epi means on top of, so it's also called genomic imprinting. So while you go through the course of your life, every cell in your body, genes are being silenced, genes are being turned on. That's part of gene expression. And every generation, any imprinting in the eggs and sperm genome, almost all of it is erased every generation. But it has been discovered in the last few decades that some of this imprinting remains and is not erased for the next generation. And especially genes that are related to energy consumption seem to be ones that may be silenced in the next generation. So some very interesting experiments have been done with this, but there are certain genes that the paternal version of that gene is the only one that's used during embryonic stages. So, but epigenetics basically means it's an inherited indication on the DNA that a gene is either going to be shut off and is not going to be used, or it's an indication that that gene is a paternal or maternal origin gene. But almost all the imprinting that people accumulate during their lifetimes is erased in gamete formation. So we talked about at the beginning of this section about Mendelian inheritance patterns, and that we now understand Mendel's independent assortment, and his law of segregation is based on what's happening with the chromosomes during meiosis. We talked about sex-linked traits, and mostly what we're talking about when we talk about sex-linked traits is recessive alleles that are found on the X chromosome. Sex-linked traits tend to be things that we either find mostly or exclusively in boys, or things that are more severe in boys, like in the case of a X-linked dominant trait. We talked about using the linkage of genes to map chromosomes, although we're not going to do any calculations with that, but that has to do with dihybrid crosses. Changes in chromosome number or structure does cause disease, and we call that aneuploidy. Deletions, insertions, inversions, those are also major alterations in chromosomes. And even though they're usually deleterious, we can see that it has also occurred during evolution. Chromosome number has changed. Big pieces of chromosomes have been moved around, so it isn't always deleterious. Usually it is. And lastly, we talked about especially organelle chromosomes like mitochondrial chromosomes that are not inherited in the same type of inheritance patterns because they are essentially a little bacterial chromosome, and they don't go through meiosis, and they are inherited maternally. So that's the end of genetics.